Good day, everyone. We will be tackling malaria and artemisinin as our last lesson for midterms. I'm Sir Franz, and let's begin. We have here a slide showing to us the life cycle of malaria. So at the outset, malaria is a hemoprotozoan, which means that the pathogen we are dealing with is a protozoan, and it's found in the blood. Our definitive host would be the mosquito. And if we use the term definitive host, we are referring to the host that harbors the sexual life stage of our parasite. So our intermediate host is the human, which means that the human holds the asexual stage of the parasite. So let's begin. First, how does the human get infected? The mosquito, which harbors the sporozoites, takes a blood meal, which means that the mosquito bites the human. And as the mosquito bites the human, it injects sporozoites into the skin of the human. And eventually the sporozoites, by way of blood and lymph, reaches the liver of the human. So here, the sporozoites enters the liver cell, and inside the liver cell, the sporozoites will now develop into liver schizons. And as the liver schizons continuously multiply and grow, it eventually ruptures the liver cell. And as it ruptures the liver cell, it releases schizons into the blood. Now, the schizons enter the red blood cell and infects the red blood cell. At this point, we are already in the human blood stage. So, as it infects the red blood cell, like in the liver cell, it will continuously grow and multiply, and eventually, this schizon forms a ring stage. And this ring stage can cause the schizon to grow and multiply and eventually rupture the red blood cell, or the ring stage, as it has infected the red blood cell, can develop uniquely into gametocytes, or in common terms, the egg cell and the sperm cell. In humans, the male and the female gametes. So the male and the female gametes will then be taken up by the uninfected mosquito. And in the uninfected mosquito, these gametes will continue to develop into a macrogametocyte for the female and a microgametocyte for the male. And they will fuse to form the ookinete. So para silang nagkakaroon ng fertilization. And the ookinete becomes an oocyst, which goes into the gastrointestinal tract of the mosquito and eventually into the salivary glands of the mosquito, where it will release sporozoites, which are now ready to invade the human once the mosquito takes another blood meal. So let's recapitulate what is important for us, the human stages. The human stages again starts with the sporozoite injected by the mosquito, goes into the liver. And in the liver, that is what we call the exoerythrocytic cycle, meaning outside the red blood cell. The liver again harbors the sporozoites, which develops into schizons, or what we call merozoites. And the schizons then continuously develop until it ruptures the liver cell, releasing into the blood the schizons that we call already blood schizons. So the blood schizons will then infect red blood cells, form the ring stage, continuously multiply in the red blood cell, and cause a rupture of the red blood cell, or it can develop into gametocytes. I want you now to focus your attention in the erythrocytic cycle. So red blood cells get infected, and then inside the red blood cell, it multiplies and then ruptures. So that cycle of infection up to rupture has a particular time period. And the rupture of the red blood cells will eventually produce the clinical findings that we see in malaria. So these are chills, fever, and myalgia or muscle pains, and then eventually this will release hemoglobin into the blood. So that cycle of rupture corresponds to our clinical presentation. So we have, according to that, 4 clinically relevant species. Number 1 is Plasmodium falciparum, which is the malarial species of greatest concern because it causes your malignant tertian malaria, which means that it has the potential to infect the brain and even the kidneys. And this will be the point of exit or cause of death of the patient. So your Plasmodium falciparum is present, sadly, here in the Philippines. So let's look at the fever pattern of Plasmodium falciparum here. Plasmodium falciparum is described as malignant tertian fever of malaria. So tertian refers to 3, right? So that means that every third day there will be fever. So fever at day 1, then it goes down, and then day 2 enters, and then day 3 there will be fever again, and then it goes down at day 4, and then at day 5 again. So the fever intervals will be every 48 hours. So that means that every 48 hours, red blood cells will rupture. Again, it's malignant because it causes great morbidity and mortality, particularly the central nervous system, the liver, and the kidneys. So it may cause acute renal failure. Next, we have 2 species, Plasmodium vivax and ovale. So Plasmodium vivax and ovale are described as benign tertian malaria. Like your Plasmodium falciparum, the fever will occur every third day or an interval of 48 hours. It's benign because the amount of disease that it causes and the severity of the disease is not as lethal as that of Plasmodium falciparum. And what's unique with Plasmodium vivax and ovale is that it has the hypnozoite stage. Remember the life cycle where initially the point of entry will be the liver cell, and then the liver cell will rupture and release into the blood your merozoites. So in Plasmodium vivax and ovale, there can be a dormant stage in the liver, and it may go so that the patient has already been treated with drugs for blood schizons, so we have eliminated the malaria in the blood, but there is a focus of infection in the liver that, if left untreated, will result to recrudescence or infection, reinfection of the blood because there is some sort of a seed left in the liver. So we have to treat the blood and we have to treat the liver to completely eradicate Plasmodium vivax and ovale. Next, we have Plasmodium malariae, and it causes what we call benign quartan malaria. So in benign quartan, quartan refers to 4. So every 4th day, there will be a fever. Fever at day 1 goes down, day 2, day 3, no fever, and then on the 4th day, there will be a fever. So our interval for the fever, which represents the lysis of red blood cells, would be 72 hours. So now let's go to the treatment options. Here in the middle is the stage of infection, which is better visualized with our life cycle diagram. So you have infection of the mosquito, hepatocyte invasion, liver stages, erythrocyte invasion. So sasabog yung liver cell. It infects the erythrocyte or red blood cell. And then you have here your asexual intra-erythrocytic stage. And then eventually some of it will develop into the gametes, sexual differentiation. And you have sexual intra-erythrocytic stage, which happens in the mosquito, and transmission to the mosquito and by the mosquito. So what's important for us here would be the drugs. We have particular drugs that we call liver schizonticides, which means they will kill schizonts in the liver. These are your 8-aminoquinolines and your folate inhibitors. We have blood schizonticides, which infect the red blood cells, which target the infected red blood cells. And these involve artemisinins, the cinchona derivatives, quinolines, folate inhibitors, and atovaquone. And we have drugs that inhibit sexual differentiation, artemisinins, 8-aminoquinolines, the folate inhibitors, and atovaquone. Our point of interest here would be the artemisinins, which are effective in the asexual intra-erythrocytic stage and the erythrocytic stage of sexual development. Our artemisinin derivatives come from the plant called Artemisia annua, which is also known as the sweet wormwood, which has been discovered first in China. So for this, a Nobel Prize for Physiology and Medicine was awarded in 2015 to Professor Yo-Yo Tu. So Professor Yo-Yo Tu is a chemist who led a group of researchers to develop or isolate artemisinin. So again, like our discussion in the past, they started with folklore, and from folklore, kwento ng mga sinaunang healers, they were able to locate the sweet wormwood, and from the sweet wormwood, the process of drug discovery from nature and eventually isolating the structure was delved into. So eventually they were able to derive artemisinin, which has this chemical structure. So how does artemisinin work? The protozoal cleavage of artemisinin endoperoxide by heme produces toxic heme adducts. So your Plasmodium, remember, infects the red blood cells because in the red blood cells, we find their heme. So the heme is their source of iron and eventually energy. So they eat the heme found in hemoglobin found in the red blood cells. So if the patient takes in artemisinin, together with the heme, your Plasmodium will also engulf artemisinin. And in the stomach or in the digestive vacuoles of Plasmodium, artemisinin together with the heme is exposed to an acidic environment, and that causes the artemisinin endoperoxide, which is this one, to react with the heme iron producing heme adducts. So the heme adducts then are unable to be degraded. So they are not degraded, and eventually they will produce free radicals that Plasmodium will not be able to eliminate. And those free radicals will then cause oxidation of macromolecules in the Plasmodium. So that involves oxidation of proteins, oxidation of nucleic acids, oxidation of lipids. And eventually that destroys the cellular structure of Plasmodium, causes the Plasmodium to die. So it's good to know the mechanism of action. Lastly, we have here the derivatives of artemisinin, which are products of further manipulation of the structure of the artemisinin molecule. We have dihydroartemisinin, artesunate, and artemether. Of particular importance, these are water-soluble, which means they can be taken orally. So we also have combinations of artemisinin derivatives with other agents that act by different mechanisms. You have artemether and lumefantrine, artesunate and mefloquine. Mefloquine, again, is a cinchona derivative, right? And you have artesunate and the folate inhibitors sulfadoxine and pyrimethamine, and dihydroartemisinin and piperacine, which is an alkaloid. So that's it for our lecture. I hope you were able to refresh your understanding of our protozoal infection, the most important protozoal infection of man, that's Plasmodium infection or malaria. I hope you were able to recall the life cycle, particularly the liver and the red blood cell stages, and understand that artemisinin targets the red blood cell stage. So artemisinin was discovered by a scientist from China who won the Nobel Prize, and eventually we were able to improve on their discovery and develop these other derivatives, dihydroartemisinin, artesunate, and artemether, and eventually use them as combinations. Whereas here in the Philippines, our go-to drug would be coartem. So that's artemether lumefantrine for the treatment of moderate to mild Plasmodium malaria, Plasmodium falciparum infections, which are endemic in Palawan and in Davao del Norte. So thank you for listening. See you next time.